So I'm grateful to the organizers for putting this talk after the end of the World Cup. Thanks for coming. There's a lot of parallel sessions going on everywhere. So this is a paper that we never intended to write because the idea was to characterize the intrinsic biases of DNase I and then go on and find out things about cofactor combinations from in vivo DNase data. But somehow it took us about 3 years to get a null model for DNase I, and we discovered all kinds of interesting things, not about DNase I, but also about possibly a new way in which cytosine methylation in DNA can be read out and modulate the strength of protein-DNA interactions. So this is a collaboration between my lab at Columbia and then, this is not working, I think, but is there a backup? Sometimes you have to shake it. It's very, I'll change the batteries as I talk. So it's a collaboration with Remo Rose at USC, who is a computational structural biologist who does supercomputer simulations of DNA molecules. And it was an important part figuring this out. And then John Stam in Seattle, who is doing a lot of DNase I digesting in his lab, in the context of the ENCODE project. So, this is the wrong size battery. I'll give up on the pointer, that's okay. Just a little bit of background, DNA, so it's a footprinting agent. It has, of course, an endogenous function in the cell, but great, thanks. Where do I push? The green thing? Okay, great, oh, perfect, great. So just very quickly, you can use DNase to do footprinting classically in a gel, which has been done for a long time. This is a very widely used technique. And then more recently, it's been scaled up in combination with high-throughput sequencing to do footprinting of chromatin at all kinds of levels of resolution down to individual footprints, binding sites of individual transcription factors, right? And then this data is interpreted in terms of, system regulatory function, et cetera. So if you look at, in the Manianis book, Molecular Cloning, in the appendix about DNase I, it says, there's now a 4th edition, but in the 3rd edition it says that the intrinsic specificity of DNase I is a surprisingly murky topic with conflicting, evidence from different groups. Turns out there's like 2 15-year-old papers that kind of contradict each other, and then people gave up. One of the papers is actually right, the other is wrong, we found out. If you read our paper, you'll know which one. But there was also a reason why we were interested in DNase I, and our lab is interested in modeling protein-DNA interaction quantitatively in general. We've done some work on CellX-seq with Richard Mann, like using high-throughput sequencing to profile protein-DNA interactions. We've developed algorithms named reduce and matrix reduce and feature reduce that analyze PBM data. And so DNase I is very special DNA-binding protein in the sense that it in a way, says thanks by cutting the DNA after it's bound somewhere. So that's great because you get a single base pair resolution mark of where DNA was bound on the DNA. And if you assume that it's going off and on the DNA a lot of times before it cuts, basically what you're sampling is the equilibrium binding behavior of the DNase I protein on the DNA, but you're reading it out through these cuts, which are single-stranded cuts, by the way, that it leaves. It cuts out one of the phosphates, one of the strands of the double-stranded DNA that it binds to, right? And so we were interested in this as a test case for looking deeply at the sequence specificity of a DNA-binding protein. Now, if you have data where you have like tens of millions of cuts across the genome, and here it's naked DNA, so it's DNA purified from a cell line, but then it's treated with DNase I after all the proteins in the chromatin has been removed, right? So we're just looking at how DNase cuts naked DNA. And then every time it cuts somewhere, we're keeping track of the local sequence context. For instance, the number of cuts that are made, this P is the phosphate that's cleaved out, right? In the context of an ACT-TCA hexamer, right? There's 7,100 cuts made in this data set in that kind of sequence context. The number of genomic positions is, varies with the hexamer, so you have to control for that, basically by taking the ratio, it turns out, right? And then, of course, this ratio, because it's observed number of cuts divided by the genomic positions, scales with the total size of your library. So it's not interesting per se, you want to rescale it, and it makes sense to rescale everything from 0 to 1, right? So the most cleavable hexamer, the one that's cut at the highest rate, an individual site in the genome with that hexamer context, right, is on top of this table, and then it goes down, and every hexamer has a score between 0 and 1. Well, if you plot the distribution of those scores as a cumulative distribution function, right, where the peak is somewhere here at the inflection point, this is a little small, but it goes over 3 orders of magnitude, from 10 to the minus 3 over here to 1. Right? So there's a dramatic dependence on sequence, goes over a factor of 1,000. The most cleavable place in the genome and the least cleavable place in the genome, their cut rates differ by a 1,000-fold intrinsically, which is a property of the DNase I enzyme. Now, since the reason why we looked at hexamers is that we did some prior analysis, like how far we think the influence of base identity extends, and that was a reasonable range that also is consistent with the crystal structure of DNase I bound to DNA. But what is nice about having an accurate estimate of a relative cut rate or binding affinity, equivalently for every hexamer, is that you can actually analyze dependencies between different positions within the same hexameric binding site in a very explicit way. So what this plot shows over here is every point is one hexamer, but we're doing something special at the first position, 5' to the left of this cut phosphate. We're considering cases where you compare the same sequence, but then one with a T at that -1 position to one with a G at the -1. So what's the effect of mutating a T, just 5' of the cut site, to a G? And then there are 4 to the 5 remaining, combinations for the ends over here, right? So there are 4 to the 5 points in this plot, right? Now, if the effect on the binding energy, due to this mutation from T to G at the first upstream position is independent of what's going on at the other positions, you expect these points to be on a straight line, right? The ratio y over x for every point is the same, right? It's the same fold change in cut rate or affinity, which corresponds to an additive effect of the binding free energy from that base pair or nucleotide position, right? So in this case, it's true, right? All these points, they could be summarized, their y over x ratios could be summarized pretty well by a single slope, which is this dashed line that I showed, right? But if we go to another position, say the second downstream position, we consider changes from an A to a C at that second downstream position, suddenly there are 2 subsets of points. There's basically points that lie on the line with the slope of 1 that are insensitive to this mutation. They don't care going A to C at +2, right? And there's points that actually do care about going from an A to a C. And if you look at the actual sequences, and there's a pattern there, and I'm coloring these points, these 4 to the 5 sequence contexts, in terms of the adjacent base at the +1 position, which if it's a T, there's something special going on. Then it cares about this mutation from A to C at the adjacent position. But if it's one of the other 3 bases there, it doesn't care. Now, so this is a clean way of looking at dependencies, but also it gives a hint. Whenever there's dependencies between neighboring positions, you start to think about DNA structure or flexibility or stacking of base pairs and things like that. So that got us on a quest to look at the fine detailed structure of B-form DNA, where you worry about the precise geometry of the grooves of the DNA to understand this phenomenon. Right? Just I showed this for one particular case of a mutation position at some position and then keeping a base at some other position fixed, right? You can do this globally, right? You mutate the position at position J, a base at position J, and then you keep some other base fixed at a different position J prime, right? And then you can do a weight matrix-like approximation where it doesn't depend on this context, right? If you take the difference between these 2 matrices, and these are the changes in binding free energy, so it's delta, delta G, right? Delta G is bound versus unbound. Delta, delta G is the difference between 2 different DNA sequences of the delta G's for use. Now, if you take the difference between 2 delta, delta G matrices, you get a delta, delta, delta G matrix, which is over here, right? And what you see is that most of the dependency is actually close to the diagonal, so they have to do with stacking indeed. And globally, that's also true. Okay, so now, we were trying to understand, in terms of a structural mechanism, what is going on here. So in order to do that, Remo Rosa's lab performed all atom Monte Carlo simulations of free DNA in solution. It was actually using an implicit solvent model for this, right? And then that would give us predicted shape parameters, things like minor groove width and the role between adjacent base pairs from these computer simulations. But they had nothing to do with DNase I or the kind of experiments that we're trying to explain, right? And what we found is that there are very strong trends between shape parameters, in this case like minor groove width, at a particular position within the binding site. This is the leftmost position, right? And the cut rate, which I'm here showing as the log minus the log of the cut rate, or the delta G, right? And so, because there's these trends, it made sense to build a multiple regression model where we're trying to predict, the cut rate, or the log cut rate, the binding free energy, in terms of these shape parameters at different positions across the binding site, either minor groove width or the role between adjacent base pairs. Right? And no, that worked reasonably well, but we made another observation, and that is when we compared our Hexamer model derived from yeast digests and digest of IMR90 or human cell line, and since it was the same bovine cow DNase I enzyme and we were controlling for the composition of the genome, we expected this plot to be very tight, along the diagonal, like a comparison between two replicate experiments would be, which is shown in this inset. But it was actually not the case. There was a lot of scatter, and when we looked at this a little more closely, we found that these points that form a separate diagonal, they all share the property that they were cuts made right adjacent to a CpG dinucleotide. So then CpG, you think, hey, maybe it's methylation, but yeast doesn't have methylation, but human does, right? So maybe DNA methylation has something to do with this inconsistency between the model built from yeast and from human. Now, this was, took a couple of years to figure all this stuff out. So in the meantime, the Lister et al. Paper where they did the methylome bisulfite sequencing, of all cytosine methylation status of the IMR90 fibroblast cell line came out. And then John Stam's lab for this project did a deep digest of naked DNA isolated from the very same IMR90 cell line. So we had parallel DNase, naked DNase data, and methylation status data. And so that allows us to partition the genome into high and low methylation regions, or actually at the level of individual base pairs. And then we could build separate cut rate models for the low methylation and the high methylation part of the genome. And now you see the same pattern as before between yeast and human, but much more cleanly. All these points over here, they are adjacent to CpG. These are all the other sites. They're on some diagonal, right? So the ratio of the slopes of the red points here and the other points, that's basically the fold effect of CpG methylation on the cut rate. It depends. It doesn't really matter what the sequence is, but if it contains the CpG, then between the unmethylated version of that hexamer and the methylated version, there's about a 10-fold increase in the cut rate when it's methylated, which you also expect to see in all the DNase data that's collected in the ENCODE project. Actually, when we had this observation, we thought maybe we can put this in reverse. We can maybe infer methylation status from in vivo DNase data, right? So in vitro, naked DNA is a very uniform distribution, apart from this 1,000-fold difference to base, right? But if you look at hypersensitive regions in some cell system, right, in nuclei of a particular cell line, in the regions where you have local hypersensitivity, you have a pretty high density of coverage, right? And then what we did in addition, we looked at windows of 1 or 2 kb, right? And there were several CpGs in those windows, and then we compared the expected number of cuts with the observed number of cuts. And that way we could build some kind of index of methylation locally. So when we did that, on the one hand, we predicted a bunch of sites that we thought were low methylation based on this insight into the biases of DNA. Methylation depends on DNase. And then this shows the actual distribution observed in the methylome data according to Lister et al. So you see that indeed, the ones that we predicted to be low methylation from only the DNase data tended to be pretty lowly methylated, and then these are predicted high methylation regions in the genome, and those tended to be well above average. I'm not sure that this is the way you want to do the profiling, right? Because you can only do this in a relatively small subset of the genome where you have high enough coverage. But with increasing sequencing capability, at some level of resolution, you can get methylation information for free from DNase data, which may be interesting depending on what you're working on. Now, so we had made two observations at this point. On the one hand, we had seen that depending on the local DNA sequence, there's a 1,000-fold variation in how happily DNase will cut this single-stranded DNA there. And on the other hand, there was like this 10-fold dependence on methylation. And so we figured, since we already had some clues that DNA shape, the role or the minor groove width might be really what's determining the binding affinity between DNase and DNA, and then the rate at which it would be cleaved, we thought maybe it's the same thing. Maybe these are just two ways of changing in a subtle way the geometry of the minor groove of the DNA, right? So if that's true, maybe we could train a model only based on the unmethylated data, right? Train a model to go from shape parameters to a cut rate or an affinity, right? And then if we would then plug in the predicted changes in the DNA shape due to methylation, we could propagate them through that model that was built from the unmethylated DNA. Maybe we could reproduce this finding that we observed that methylation enhances the cut rate. Right? Now this shows for the same sequence, so this is a simulation done by Remo Rosa's lab. Red is the unmethylated version of the DNA, and what you see here is that when you methylate the cytosine, the two cytosines in that sequence that contains the CpG over here, right? That upstream, which is closer to where DNase actually goes into the minor groove and cuts the DNA, that there is this narrowing of the minor groove by on the order of an angstrom. Now what happens is the phosphate residues on the backbone of DNA, they're negatively charged. And if the minor groove narrows, they're closer together, they create a deeper electrostatic pocket that's then more favorable for arginine side chains, which is basically how DNase recognizes the minor groove to go in. So it's an enhanced electrostatic interaction between the positively charged residue, like arginine in particular of DNase I, and the negative charge of the backbone. And because of this, and a lot of transcription factors actually have contacts between arginines and the minor groove in crystal structures, we're now actually exploring the idea that maybe this could be a much more general mechanism that could occur in the context of regular transcription factor binding, and we're kind of in full swing trying to show this in different ways. Right? But now, so this is pure DNA structure simulation, but now how about using this change in DNA shape to predict the change in DNA's cut rate or binding affinity, using this shape to affinity model that we trained on the unmethylated, the red type sequences, right? And this shows actually what happens there. So again, we built this model from unmethylated cut rates only, the numbers on this axis, right? And then we used the supercomputer, prediction of the change in shape to predict the cut rate for the methylated version of the same sequence. So every point is a sequence that contains a CpG downstream of the cut, right? So there's 4 to the 4 sequences in this plot. The different colors are different types of models. The model that did not work well to predict the effective methylation was actually a model based on minor groove width. So these blue points, they're on the diagonal. The model doesn't predict any change in the cut rate due to methylation. But if we included the roll parameters between the different neighboring base pairs in the model, suddenly the predicted delta G for the methylated version was lower. See, these points are shifted down from the diagonal, right? So you start with some affinity or log affinity, and then you go down when the DNA is methylated. Right? The highest, most cleavable site is here at 0, and the weaker, the ones that are not as cleavable, have a more positive delta G. And a shift of the log is corresponding to a scaling factor on a linear scale, right? So the same thing with the two diagonals, methylated and unmethylated, is confirmed by this. But this is purely predicted now from our model of unmethylated DNA and the change in the shape of the DNase parameters. And actually, if you take this shift and you exponentiate it, right, e to the power whatever this is, that is very close to within like 10 or 20% to the observed effect on cut rate in the DNase when we use the Lister data to partition the genome into high and low methylation. So it's showing qualitatively in a way that the effect of methylation is independent of the sequence contact, because the shift is independent of what sequence and what cut rate you start from. And then also quantitatively, the amount, the change in the delta G is also is in the same ballpark with a relatively simple model, which is nice. So just to wrap up, we've shown these strong biases in DNase I. Shirley Yu and Miles Brown had a paper, and Shirley presented 2 days ago about this, where, this specificity, depending on the context, in particular when you use motifs, and then do aggregation based on motifs, you have to be a little careful in interpreting your in vivo data. It definitely makes sense to take this model into account. And actually, it's surprisingly hard to do this, beyond just saying that there is an effect. And we're working on that to basically, incorporate this in footprinting methods. Right, and the other thing that it's all related to this structural mechanism related to the minor groove. And then the other observation was that there's this tenfold effect of CpG methylation, which we think could be more generally important than just for this DNase enzyme, and we're working on that. So with that, I in particular want to acknowledge Alan Lazarevic, who was the first author on the paper, and then again, like Remo Rose and John Stam, also Todd Riley in my lab was involved. And we developed this very much in parallel with the select-seq methodology, so there was a lot of cross-fertilization between this and our models we built from the Hox data in the context of that paper with Richard Mann. Thanks for your attention.
Thank you. Beautiful talk. Please.
Hi.
Can you present who are you so we know?
Sure. Ferhat Ay from University of Washington Genome Sciences. Great talk. Thank you. One question that I had was all these bias analysis that you are doing is at a certain DNase concentration? All the samples are coming from the certain, whatever the standard protocol is?
It's a good question. Strictly speaking, I think depending on the experiment, there's some tuning of the conditions that's being done, right? So if you want to get really quantitative about this, or you could argue that you have to do a separate control for every experiment. The hope is, we're just the fact to show that there are these strong biases, right, for a particular condition. It was mostly our point and this insight into methylation. But I'm sure also depending on like the cation concentrations and whether it's like manganese or magnesium, the enzyme can operate in different modes, even like single-stranded versus double-stranded, and so all that stuff. You could vary systematically in principle and see what happens, how it affects.
I'm wondering because if you take it to the extremes, use a very high concentration of DNase I, then maybe like it would have the sonication effect, right? It would randomly digest DNA all over the place, right, versus in here, there's a very light concentration. Of course, it wouldn't be useful for this DNase assays where you are trying to find the open chromatin, but if you just want to do something like sonication with DNase, maybe that's achievable. Right. Yeah, that probably.
By the way, we looked at sonication bias as well as part of this study. There's a 30-fold depends on the local dinucleotide context of where the cut was made, even like from SonoSeq data that we analyzed for that.
Yeah. Thank you.
Hello, Yulia Sitnikova from Brandeis University. My question is, did you check hydroxymethylcytosine? So is it also given the same effect?
No, it's an interesting question to what extent like other modifications, right? And also there's a lot of adenine methylation, like in bacteria, etc. We haven't really systematically pursued that, and also, around the time of we were wrapping up this study, there was not yet like high throughput hydroxymethyl data. Otherwise, we might have considered that. We were aware of that, but I think it would be interesting, for other people to look into this for sure.
And did you check it in CpG content, in CG dinucleotide content? Because you have one strand methylated in one place and another strand in another place.
Right. So it's the cut that's made 5' of the cytosine on either strand, because CpG itself as a dinucleotide is symmetrical, but it's the cut made 5' of the C that's enhanced by the methylation.
From both sides, it will be.
Well, from both strands on the 5' side, but not on the 3' side.
Okay. Great. Thank you.
Hi, this is Jiang Chen from Johns Hopkins. So DNase I HAP-sensitive size profiling is usually to identify the active regulator region. And on the other hand, the DNA methylation often associated with the inactive regulator region. So your data is suggesting that the DNase I profiling actually include a lot of false positive? And if that's the case, is there a way to normalize this effect?
Well, yeah, so I think that's another thing that could be useful. We haven't, because we're, again, trying to focus on the mechanism, but it is conservative in the sense that to the extent you believe methylation shuts down transcription, which, the typical like MEBP2 recruiting an HDAC, right? Which is only a subset of what happens. I think, it could also go the other way. But if you think that methylation shuts down transcription, then that you have less open chromatin as you get a lower signal, right? Even though intrinsically the naked DNA would get more cuts, right? So it's harder in a way for that region to show as like a closed down region. So in that sense, it's conservative, that expect false negatives rather than false positives. So if you incorporate this methylation enhancement into a model for detecting hypersensitive regions, you might find more hypersensitive regions.
Because in the ideal case, we should expect that the DNase I cleavage only depend on the chromatin state, not by other sequence preference or DNA methylation status. So given your data, that do we need to do some normalization based on the genomic sequence?
It's when you look at single base resolution, it's definitely a good idea to take this into account. Again, like how exactly you do that, it's not a trivial problem to solve. On the other hand, because if you move by even one base pair, the local sequence context and the chemical context changes completely, right? So as soon as you compute like sums over windows of even like 24 base pairs or longer, it kind of self-averages. So the expected cut rate is like the genome-wide or the average, across the genome times the length of the window instead of summing up our model. We tried the footprinting sensitivity in a way using our model and not with windows of say 100 base pairs. And it was hardly a difference in terms of what regions you would call hypersensitive or not. So in that sense, that is pretty robust, right? If you look at single footprints, you may have to be a little careful interpreting the fine structure of that.
Okay, thank you.
You basically answered my question with that.
Okay.
Maybe can I ask you?
Sure.
I mean, everything you said was for naked DNA, basically. But in reality, there is a lot of linking number and supercoil, supercoil, and so on. How the topology of the DNA is part of the game?
Yeah, that's an interesting question. I guess you could create like circles of DNA with a particular linking number or something, and then digest them and see how they. And maybe there's, we did a lot of like mining of like 15, 20-year-old literature. Maybe I'm sure that there's a good chance that people have looked at this as well. It's a very interesting question. Yeah. Yeah, and maybe you could even be a signature of chromatin like Lucas. Yeah, it sounds great.